be doing, we're good extension people, so we will be doing a survey and an evaluation, and it's going to be completely computer evaluated. You know, if you say you have 5 hives, Bill's not going to get to see that, to see how many, you know, there's no names involved. I just wanted to make that known. Our next presenter is kind of a brand new Texan. We're really tickled to have Dr. Juliana Rangel here to talk to us. If you look at her resume, she's come out of North Carolina and has a world of research backing up her interest in queen genetics. And so we're pleased to be one. Are we your first Texas?
Yes.
Very good.
Thank you so much for the invitation. Fred contacted me last year when I hadn't started my job at Texas A&M in College Station. So this was my first official talk to a beekeeper group in Texas. So I hope I don't disappoint. But started my new job about a month ago. So I'm brand new to Texas. How are you guys doing? Howdy.
Great.
Howdy. As an Aggie, you have to say howdy every time. Good. So I conducted a postdoctoral fellowship study at North Carolina State University with Dr. David Tarpey. He's interested in queen biology, and so am I. I got my PhD at Cornell University, and I was originally working for my dissertation on the biology of swarming in Apis mellifera. And then I have broad interests in reproductive biology, so I moved on from swarming for now to queen health, queen quality, and hopefully queen supersedure once I get the event of supersedure to happen when I want it to happen. Because you know when you're looking for something, you can't find it, but if the moment you stop looking, then you start seeing it. So we have big problems with supersedure in the beekeeping, large and small beekeeping operations across the country. People are complaining about poor queens and poor queen quality and supersedure a lot. We're in the process of getting to understand the process of supersedure, what causes it, what workers do in order to replace the queen, and so on. And so I'll be talking about a few studies that I've been working on queen reproductive biology, and then at the end I'm going to give you an outline of my broad interests at Texas A&M, what my job responsibilities are, and just so that you know that there is now a bee biology laboratory that is starting off, but that has great potential at Texas A&M. And my goal, even though, as you will see, my job description does not contain any extension, it's mainly research and teaching, I do know that a good research program, it gets even better with the help of the local beekeepers in wherever state you are. So if you have any questions, concerns, ideas, please email me, and eventually with time, at this point there's only 3 of us in the lab, but there will be hopefully a lot more, and students, and so once we get students involved, we will have, if I cannot attend meetings, I can probably send emissaries with information to talk to the beekeepers about. So the outline for today's talk is, it's quite simple. I'll start off with some basic biology of queens as a refresher for most of you, as new information for some of you, but this will tie into why I'm interested in the studies that I will be talking about. So I have data, actually quite new data. The first project that I will be talking about is the effects of queen reproductive potential or queen quality on colony growth and supersedure, and that was published late last year. So I have the paper. If you want me to email it to you, I can send you a PDF of it. This one is actually in, it has been submitted to a journal, so it's quite fresh. No one really has seen this information, but it's in press, so it should be published sometime this year. The effects of commonly used miticides on the mating health of honeybee queens. And then I'll finish off with some of the items of my, my research interests at Texas A&M. Okay, so let's start with regulation of queen production in honeybee colonies. As most of you know, the honeybee queen is the mother of all the members in the colony. She is the breeding depository of all the inherited characteristics of the colony acquired from her progenitors and from the males that she mates with. So she provides half of the genetic material to all the workers and drones that she mothers inside the queen. The other half comes from the drones, so that's a very important part of the equation. As you will see later on, I am very interested in learning about drone quality, not just queen quality, so we will be working on drone quality as well in my lab. The queen is also the only female that reproduces and is capable of laying fertilized and unfertilized eggs. If she's properly mated, she will lay up to 1,500 to even 2,000 eggs a day. She is larger than workers, and her abdomen is elongated because she carries millions of sperms and eggs in her ovaries, and the sperm are collected inside a special organ called the spermatheca that contains millions and millions of sperm cells that she then uses one at a time if she decides to fertilize an egg, and then it'll eventually turn into a worker, of course, and if the egg is unfertilized, it'll turn into a drone. So here's a queen laying a little egg. A queen-right colony, that's what beekeepers call it, a queen-right colony is a colony with a functional laying queen, and it functions largely as a cohesive unit because of these pheromones that she releases, mainly from her mandibular glands. So they're called queen mandibular pheromones, or QMP. QMP is basically a blend of chemicals that she releases, and it tells the colony that there is a queen and that she's there and laying eggs and she's mated. And so that QMP is transmitted throughout the colony between the queen, from the queen to the workers by direct contact between the workers and the queen. So these workers will come near the queen, lick her, and get those molecules of QMP. QMP is a blend of at least 5 different chemicals. Those are the main ones, the ones that we as beekeepers kind of have heard about, the 9-ODA, 9-HDA, HOB, HVA. But in reality, QMP is a blend of dozens of chemicals. And each queen has a different recipe of her QMP profile. Each queen has different concentrations of all of these dozens of chemicals, and that's what makes her signature. And that's what allows workers to know that the queen that they are around is their queen and not a queen from another colony. That's also why beekeepers, when you're requeening a colony, you have to cage the queen for a few days so that the workers don't kill that foreign queen, because her QMP profile is different and it smells different to the workers. And so they know that that's not their mom, so they have to get used to the QMP of the new queen before you uncage that queen. So it's produced by the queen's mandibular glands, and it's spread all over her body, and it causes behavioral and physiological changes to colony members, and I'll talk about those. So, we already wrote that. So this is kind of a description of how QMP is moved about in the colony. The queen releases QMP, and through her own grooming, she will kind of spread molecules of QMP around her body. And then you have these retinue workers. Retinue are the attendants, so the workers are always around the queen. Those retinue workers will, again, with their antennae and legs, will lick the queen and collect molecules of QMP from the queen. They will in turn groom themselves so that they get most of the concentration of the QMP molecules on the tip of their antennae and their mouth parts. And then they will do, during food sharing or trophallaxis, they will move those molecules of QMP to other workers. such that at any given moment, a worker does not need to come in direct contact with the queen in order to know that she's there and that she's alive and laying. All they need is to get some molecules of that QMP from other workers for them to know that that the colony is queen right. So that's how it works. Not every worker touches the queen in this way, and they still know that there is a functional queen in the colony. So that's what we call retinue response, when you have the queen being groomed or attended to by the retinue workers, so all of these attendants around her and licking her, giving her some food samples and whatnot. So when there have been experiments on QMP, you can take a queen, kind of dissect her mandibular glands, extract all of those chemicals from the mandibular glands, and then put them on cotton swabs or filter paper. You will see aggregation of workers toward that QMP because it's highly attractive to workers. So there are very important functions of this QMP. There's two main ones. One of them is that it inhibits worker ovary development. We know that workers are what is known as facultatively sterile. That means they can, under certain circumstances, activate their ovaries and lay unfertilized eggs. And that's when we have laying workers. Some of these workers will activate their ovaries and lay unfertilized eggs. However, QMP inhibits worker ovary development in most workers inside a colony. Here are pictures of, here are the ovaries of a worker, the ovaries of a virgin queen, and the ovaries of a mated queen. So as you can see, there's a high difference in size between the ovaries of a functional queen and the ovaries of a worker. You can see it better here. Normal workers usually don't have activated ovaries. Here are the activated ovaries of a laying worker, but they're still smaller than the ovary size of a virgin queen. But in any case, QMP inhibits worker ovary development, and it also inhibits queen rearing. So that in most cases, under most circumstances throughout the year, a colony only has one queen. Because the queen mandibular pheromone blend is also telling workers to not rear queens. So the nest is almost always having these prepped queen cups just in case that queen rearing needs to get started. It already started the process with the queen cups or starter cells to raise queens. In the presence of QMP, they remain unoccupied, but in the absence of QMP, that is either if the queen is dead or if there is crowding inside the colony and there's really no way for those workers to transmit the QMP to the other workers, and so in a sense, some workers feel a sense of queenlessness, then the workers will start rearing queens. So they will start raising several queen cells in those starter cups that I told you about. So the two main functions of QMP are inhibiting worker ovary development and also inhibiting queen rearing. However, there are certain conditions under which queens are reared inside a colony. There's three main ones. Swarming, which is how a colony reproduces itself, is the process by which a colony divides into two units, and the swarm, what we know as the swarm, lives with the mother queen and approximately 3/4 of the worker population in a swarm. So back in the parental nest, that quarter of the population that is left behind needs to have a queen. So the colony will have started queen rearing before swarming so that at least one queen will inherit that old nest before the swarm leaves. The other situation is emergency queen rearing, and that is if the existing queen is killed or if she disappears for some reason. The colony is kind of against the clock to use appropriately sized larvae, of fertilized larvae, to raise as a new queen. And I'll explain this later, but sometimes the colony will not have the larvae of the appropriate age if the colony goes queenless for any reason. And even if there are larvae of fertilized larvae, they can't raise that larvae as queens, and so it becomes what is known as hopelessly queenless, and you've probably heard that as a beekeeper. Usually emergency queen rearing cells are located in the middle, in the center of the brood nest, and not in like swarming, which occurs usually in the periphery of frames. And there's also fewer emergency queen rearing cells than there are for swarming usually. Of course, we should put, after every sentence in beekeeping, we should put usually. Because there's always exceptions to every rule, and the bees do not go by the book. And then the third scenario is supersedure. So somehow the workers can assess the quality of that queen and or the brood. I was talking to Jerry, we still don't know what it is, but somehow workers can detect that the queen that they have is failing in her egg-laying duties, or her condition or health is not as good, and so they choose to replace her with a new queen. But the mother queen is still alive. So it's a really interesting process where there's a situation where you have two queens, and at the end, the mother queen is usually killed, and the new queen, hopefully more vigorous queen, will take up the egg-laying duties, and hopefully this will be a way for the colony to rejuvenate itself and to increase productivity. But again, as I said earlier, supersedure is a relatively understudied phenomenon, but more and more so we hear problems with queens and poor queens across the beekeeping community, and so it is a real problem. Back before we started seeing these poor queen problems 10 years ago or so, queens used to last, and they still can last from anywhere from 2 to 3 years. But nowadays we have beekeepers that purchase queens, and within the first 6 months of having obtained that queen, she will get replaced by with a new queen by the workers, which is costly to beekeepers because you invested some money in those queens that you wanted to use, but it also changes the genetic makeup of the colony. And if you wanted to use the specific queen with specific genetics, if they replace that queen, they will choose at random whatever larva they want to raise as queens, and so you will have lost that queen that you really liked for whatever reason. So that was a kind of a brief introduction to queens and how queens are produced inside a colony. I know there are other scenarios where you can have 2 queens at a time, and we don't have to go into that because, as I said, those are kind of more unique situations and not the norm. But what I just told you is like the basic biology of queens and queen rearing inside colonies. So now I'll talk about this study that we did on the effects of the reproductive quality of queens on colony growth and queen supersedure. And I put queen supersedure kind of, I should put it in parentheses because, as you will see, we really haven't been able to induce supersedure or see increased levels of supersedure under experimental conditions, but I will mention those later. So I'll walk you through this scheme right now. But there has been several studies over the last 40 years that look at queen quality. So this is nothing new. There have been a lot of studies on queen quality. And over the years, we've kind of broken it down into short sentences that one can understand better. And I'll try to explain what this means. There is plasticity or flexibility in the developmental fate of larvae within colonies. Fertilized eggs, well, a queen's fate is not determined by genetics, and I hope you know that as a beekeeper. A queen's fate is determined by what? Huh? What she eats, exactly, nutrition. So that's what we call flexibility, because depending on what she's fed, that larva is fed, it will either develop into a queen or it will develop into a worker. So there's no queen-destined egg. It's based on what she eats. But it's also when she eats that food, which is royal jelly, right? So Here we have at the bottom kind of a diagram showing the age of the larvae when initially raised as a queen. So we know that the fertilized egg, today it's in the egg stage for 3 days. And then it hatches. And for queens, it stays in the queen pathway. Well, no, actually, it's the same for both. But there's 5 days of larval development, or 5 larval instars. Okay, so these are the 5 larval days. Depending on what you feed that are fertilized larva, it will turn either into a queen or into a worker, is what I just said. So there's kind of like an intercast line right here. Research has shown that the longer you wait to feed that larva royal jelly, or the workers take, however, the longer the workers take to feed royal jelly to those larvae, the lower the reproductive quality of that resulting queen is going to be. So it drops dramatically. And furthermore, there's something called the critical age. So during the first 3 larval days of that queen, or actually of that larva, if that larva is fed royal jelly, it will develop into a queen. After the 3rd larval instar, or the 3rd larval day, no matter how much royal jelly you feed the larva, it will not turn into a queen. That's why if you, in your colony that went queenless, only have 4 or 5-day-old larvae, no matter how much royal jelly there is, or the workers feed that larva, it will not turn into a queen. Its developmental fate has already been determined, and it will turn into a worker. So there's this critical age. But even within these 3 days, that quality of the queen, the longer they wait to feed royal jelly to that larva, the lower the reproductive quality of that resulting queen is going to be. So I said there is plasticity or flexibility in the developmental fate of larvae within colonies, because they can either turn into a worker or a queen based on what they are fed. But based on what I just showed you earlier, there's also evidence that queens raised from older larvae, 3-day-old larvae for example, have lower reproductive potential compared to queens that are 1-day-old larvae when they get fed royal jelly. So there is this wide range in the reproductive potential of honeybee queens, with queens that are raised from older larvae having lower reproductive potential and vice versa. So how do we know what reproductive quality or reproductive potential is? There are several measures. Some of them are non-destructive. It's basically what we do with live queens. We grab the queen and we weigh them, and then we take measurements of their body size. So head width, thoracic width, abdomen length, wing length, and all of these things are non-destructive. You can put the queen back into the colony and she can go about her daily routine. However, unfortunately, to do the studies we do, we have to sacrifice the queen, because we have to open her up and look at other aspects of her reproductive organs, such as her ovaries, ovarials, her spermatheca. So we can determine how many ovarials that queen had, what the size of her spermatheca was or is, and especially how much sperm is stored in her spermatheca, and how viable that sperm is, because not only queens might be undermated and have fewer sperm cells, but also some of the sperm cells inside the spermatheca, for a reason that we yet don't know, get killed. So a lot of that sperm, even if she has millions of sperm, a huge proportion of that sperm can be dead, which means that she cannot use it to successfully fertilize an egg if she wants to. And also measures of mating frequency, basically how many mates she mated with, how many drones she mated with. And some queens mate with more than others, and it is known that queens that mate with about, I don't know, a cutoff point is maybe 10 mates, and if she mates with fewer than 10 mates, we say that she's poorly mated, and so hopefully she'll mate with the average, which is about 15 for Apis mellifera. Okay, so the question is, does honeybee queen reproductive quality affect colony growth and colony supersedure rate? Queen supersedure rate. So in order to do this study, we went in the spring and fall of 2010 in Raleigh, North Carolina, and we reared experimental queens by grafting. How many of you have heard of grafting? Okay, grafting is the physical transfer of fertilized larvae into a special queen cup, plastic queen cup. It can be, well, it's actually an artificial cup that you will then put into a special colony that has a lot of nurse bees so that they will take care of that grafted larvae and they will raise that larva as a queen. So you as a beekeeper do the physical transfer of the larva from a worker destined cell into that cup, and if the conditions are right, you will be able to get a queen out of that graft. So we reared experimental queens by grafting. Now, these are the 5 larval instars of a fertilized egg. So it goes from 1 to 5, or the days of larval instar. So day 1, 2, 3, 4, 5. I told you the cutoff point is about 3 days, so you will not be grafting any queens in the last 2 larval instars. In fact, you get the best, if you kind of understood the diagram I gave earlier, you get the best quality queen if you graft from the first day of larval development of that fertilized egg. And then you start losing quality really quickly as you choose older larvae. So when you're looking at a frame like this, you start looking at the larger larvae, and those are not the ones that will end up becoming good quality queens. They will become less lower quality queens. And so we reared high quality queens from 0-day-old larvae, and we reared low quality queens from 2-day-old larvae. So you can either do it like 0 to 1, 1 to 2, 2 to 3, basically is how I'm trying to say it. So from 0 to 1 days old, from 2 to 3 days old. So high quality queens, low quality queens. And then the mature queen cells, so we allow the workers to raise those cells, and they elongate the cell, and it looks just like a normal queen cell, except that it's inside these queen cups that you can manipulate later. Once that cell is sealed at the bottom, we know that the queen is now pupating, and now it's safe to move that queen cell into queenless units of workers that are ready to accept a queen. And so we place them into those new empty boxes that have approximately 7,000, I shouldn't say empty, because they will have queenless boxes with approximately 7,000 workers each. So we have 2 types of experimental colonies. We had 20 high colonies headed by high quality queens. Those are the ones from the 0-day-old larvae. And then we had 20 low quality queens or 20 colonies headed by low quality queens. We did those non-invasive measurements of those queens after we did, and I should say, we waited until those queens mated. So they came out of their cells, they started roaming around, and a few days later they went out and mated naturally. They came back. Once we knew that they had successfully mated, because we started checking for eggs about 10 days later, then we did these studies. We grabbed the queens, we did the non-invasive measures of size, and we also took some other non-destructive measures taken monthly from each colony. We wanted to see colony growth. So I should have earlier said that these colonies that had the 7,000 bees, we also provided them with empty frames that had no drawn comb. They only had foundation, wax foundation, so that they had to raise their own comb. And we provided full wax foundation, which will turn into worker comb, but they can also, we also give them alternating frames with only strips of foundation, so that they could raise or build drone comb for drone production if they wanted to, if they if they wanted to, they could raise drones as well. So we took non-destructive measures from each colony every month, looking at the amount of worker and drone comb built, the amount of worker and drone brood produced, and we looked at basically sealed brood because that's a good indication that it's very likely that that brood is going to emerge as adults. The amount of food store, and we did some estimation of worker population size. So here are the results of that study. We did notice significant, in biology everything has to be done with statistical tests and statistical cutoff points. And so when I say statistical significance or significant results, that means that it kind of passed the statistics test based on this value. So if this value of P is higher or lower than 0.05, that means that it is statistically significant. So for queen weight, we have here queen quality, low quality queens, and high quality queens. We saw that on average, high quality queens were heavier in weight. They also had a wider thorax compared to low quality queens, but we didn't see a difference in the head width. So this just really says that there are some patterns of high quality queens being larger in size than low quality queens, but other studies have also shown that queen size is not necessarily associated very directly with the other measures inside of her reproductive organs as far as reproductive quality. These are the cool ones. We followed these colonies from day 0, the day of establishment in May, all the way through the following year. And so day 0 here is the day after colony establishment. And we, at least for certain measures, we followed these colonies for about 4 months, because that's kind of the time where they build whatever they're going to use that year as far as comb goes. So in the green, we have high quality colonies headed by high quality queens, and in the in the orange, we have colonies headed by high quality queens. In the green, we have colonies headed by low quality queens. And so here we have, for example, worker comb. So the amount of worker comb, and you do that using that grid that I showed in my picture of me looking at the frame. You put that gridded frame, and that we count number of squares that are covered with whatever measurement we're taking, and then we translate that into centimeters squared. So it's kind of a standard way of measuring size, and it's done repeatedly in several studies. So we noticed that colonies headed by high quality queens produce more comb, more worker comb. And they also produce more drone comb, more than twice as much. And in this case, it's about 30% more worker comb and about twice as much drone comb compared to low quality queens. As far as worker brood production, we also find a trend, not significant, but a trend in higher production of worker brood in high quality queen colonies compared to low quality queen colonies. And we also find that low quality queen colonies do not produce any drones. They can't afford to. It's only high quality queens. Some high quality queen colonies produce or can afford to produce some drones. So depending on the quality of the queens, the colony will invest in drone production or not. As far as food goes, colonies raised by, headed by high-quality queens produce, on average, more food, more honey and pollen, so they're more productive. And they have a higher worker population size. Remember, we started off with a standard number of workers per colony of about 7,000. So these are new workers that are produced by that colony. So basically, we can say that high-quality queens are more productive overall. They produce more workers and are more productive in other ways than colonies headed by low-quality queens. Now, I don't have I can tell you the results of supersedure, and that is that between those 40 colonies, the 20 headed by high-quality and the 20 headed by low-quality queens, we only saw 2 supersedures in each treatment group. So there's clearly no evidence of higher supersedure rate based on queen quality. So we're still looking for what other aspects of the queens or something else inside the colony that causes this supersedure, but it clearly doesn't seem to be queen reproductive potential. So queens raised from young larvae showed higher reproductive quality overall. New colonies headed by high-quality queens had more worker and drone comb, larger adult population, more food stored, and more worker and drone brood. However, we did not find an effect of queen reproductive quality on the likelihood of her getting superseded. So that's the result of that study that basically is the first quantitative study of what we as beekeepers suspected, that high-quality queens produce better, more productive quality colonies. This was the first test that showed this quantitatively. So now I want to go into another study. Do you have any questions from that previous study?
Can you tell from the sperm count how many drones the queen mated with?
No. You don't do it. You do it from workers. You collect 100 workers from the colony, and you run genetic studies. You genotype those workers, and for every specific gene or part of the DNA, each drone will provide a different flavor of that gene, and so you look at that particular locus or this particular position in the DNA, and each worker that belongs to that patriline or subfamily will have that same flavor on that piece of the DNA. And so that way you can segregate between flavors of the DNA. They're called alleles, and that way you can segregate into subfamilies.
So she randomly uses sperm from all of the drones that she's making?
The evidence is that she uses sperm in a random way inside her spermatheca.
Yeah.
So
Yes.
that it's not just this one for this
There are
set of
certain bits of clumping where they can be just tiny patches. It's probably more so related to clumping of certain pieces of sperm that just clump together, and so she will use more of those in a batch of, say, 10 workers, but then she will start mixing. So it's always mixing. It might be that you can have a patch of the same patriline, but more so because of clumping than because she's selectively using sperm, as far as we know for now.
Is there any sperm degradation over time?
That's a very good question, and people at NC State are looking at that, if there is degradation of sperm over time. That's a good question. And as you will see in this study, there is something to say about sperm quality inside the spermatheca.
So I'm understanding the queen separates the sperm from different drones and some different compartments or something?
No. The opposite. It's all mixed kind
It's all
of
mixed.
randomly,
Okay.
well mixed inside. She doesn't select, or I don't think she can tell whose sperm it is and use it on purpose, or not use it. Okay, so in our quest for looking at what causes supersedure, I'm also interested in what, if any, effects there are from the use of commonly used miticides on the queen health, and supersedure. But I didn't even get supersedure with this study either. But I will show you other cool things that we found. And this is the new study. Okay, so there's current, all of these assessments of the current problems facing beekeepers and honeybees are headed mainly by Dennis Van Engelsdorf and his collaborators across the country. But in recent surveys that they've done, asking beekeepers what their best or worst problems are facing their bees, 9% problems are due to symptoms associated with CCD. 20% of colony losses are associated with varroa mites or the viruses and other illnesses that they transmit. starvation or poor nutrition, or problems with nutrition and diet, 28%, and then 31% of colony losses are due to poor queens. This is surveys done to beekeepers, so these don't have any kind of experimental design, any kind of standardization of results. It's only surveys or questionnaires given to beekeepers as far as what they think are the problems facing their colonies. And these are pretty recent surveys. So as you can see, poor queens and problems associated with varroa are pretty high up there. And poor queens is the #1 problem that they claim are facing honeybees nowadays. So with this in mind, we started working on this question of the use of miticides and their effects on queen health. Pesticides usually used in agriculture, they are used, they really help farmers and people that have huge, well, the stakeholders for a lot of very important agricultural crops. And so they use the careful use and the therapeutic dose or use of certain pesticides are used for integrated pest management paradigms, and they tend to reduce the abundance of target organisms that are becoming a pest or a problem for their crop. But there's also evidence, and growing evidence, that there are effects of those pesticides on their non-target organisms. So they not only kill the target organism, but they also affect, maybe not necessarily kill, but have what are nowadays known as sublethal effects of these pesticides on non-target organisms. And also, the target organism can develop resistance to the pesticide, tolerance to the pesticide, so even to the target organism, they can become down the road non-effective or problematic. In the case of varroa mites, we have, on the good side, we have seen a reduced abundance of the mites across apiaries across the country, and also it has become part of an integrated pest management paradigm for honeybee health. On the negative or not so positive side, we've seen the development of resistance of these mites to the miticides that we're using, and also sublethal effects of the miticides on worker health. So most studies have been done on synergisms of miticides on worker health, and a few of them have been done on queen health, but not so many. So that's why we're really interested in this. So what are some of those non-target effectiveness of drones? So it not only affects queen quality, but also drone quality for as far as reproduction goes. Similar accounts have been shown for cumaphos. Accounts for the highest concentration in parts per million in beeswax. It has shown to yield high queen mortality when the queens are exposed in low doses. So even in low doses, they have a detrimental effect on queens. Other studies, the same study showed that exposure to cumaphos during queenery lowers queen ovary and body weight, and also low queen weight and queen rearing success in another study using cumaphos during the queen rearing process. So these are scientific accounts of detrimental effects of both fluvalinate and cumaphos on reproductives in a colony. So we wanted to see if we combine the two, if we could see kind of like an additive effect or still a very detrimental synergistic effect of fluvalinated cumaphos. So we did the study last year, also in Raleigh, and we coated plastic cups with molten beeswax that was either miticide-free or contaminated with a combination of fluvalinated cumaphos. So our treatments were rearing queens in an environment that was miticide-free. I don't know why this keeps going. Sorry. Or that the molten wax was impregnated, had contamination of fluvalinated cumaphos, which from now on you'll see as F plus C. for short. But we got Burt's Bees miticide-free beeswax pellets because they use wax from, Jerry, do you know, it's South Africa?
Well, yeah,
No,
from,
Kenya.
from the Amazon and from Africa.
From Africa, so in areas where there are no mites and thus no miticide use, so we used wax provided by Burt's Bees because it doesn't contain any miticide, so that was our control group. And then our experimental group, we used that same Burt's Bees wax, but we contaminated it with the maximum concentration reported in that study of the natural amounts of these chemicals in beeswax, which turned out to be 204 parts per million for fluvalinate and 94 parts per million of cumin. So we did that in the lab, kind of melted these chemicals into the molten wax, and then we coated queen cups with that molten mixture. And then we raised or reared or attempted to rear hundreds of experimental queens using the grafting method, as I explained it earlier. We collected all the sealed queen cells, and all those mature queen cells, the ones that were sealed, were placed into new mating nukes, so queenless units of workers that will take care of those queen cells that contain only about 1,000 to 2,000 workers. And then we waited for those emerged queens to come out, mate naturally, and then we collected the queens. After we knew that they had mated, they were laying eggs, and we knew that they had mated successfully because we waited for those eggs to hatch and for those larvae to get sealed, so that we knew whether they were drone or workers. And as soon as we saw that they were worker brood, we collected those some workers from. We actually collected brood, so that we knew that those. When you're collecting workers, this is a side note. When you're collecting workers from a colony, you see a lot of drifting going on. So when you're going to do any molecular biology, any genotyping to determine what patroline or what subfamily that worker belongs to, you're going to see all kinds of crazy information, and you don't know that that worker doesn't have any of the flavors or the alleles that I told you about earlier that would place her into one of those subfamilies. And later you find out those are drifters. Those are colonies from another colony. Those are workers from different colonies. And so you wasted a lot of money on those workers. So in order to not waste any money, because it's really expensive to run these studies, doing the genetics is expensive, we made sure that those workers belong to that queen by only using brood. So by genotyping only larvae or pupae, because we know that they belong to that queen, right? So just on a side note. Okay, so what measurements did we take? We did those same non-destructive morphometric measurements that I did earlier, but then for the reproductive health part, we sacrificed the queen, we opened her up, collected her spermatheca. There's a really cool protocol we do in the lab, and then we are able to stain the sperm in a way that live sperm fluoresces under green light, and dead sperm fluoresces under red light. So that's a way for you to know whether your sperm are dead or alive. Under the microscope, it kind of looks like this, although with those filters, they glow more like green or red. And then you just take a square of standard size that you do across every queen and every sample, and then you count the number of sperms that you see on that field view of your microscope, and then you extrapolate. And so you count the number of live sperm, you count the number of dead sperm, and then you combine the two to know the total number of sperm, and so on. And so we did sperm counts. Sperm viability is basically after you add the number of live and dead sperm, combine it together to get the total number of sperm then you see what proportion of that total number of, or that total sperm is alive. And that's what we call sperm viability. What percentage of the total number of sperm in the spermatheca is actually alive and can be used by the queen to fertilize an egg? Because if the sperm is dead, then she can't use it to fertilize an egg. And then mating frequency is what I told you, how many drones the queen mated with. So, as far as morphometrics, we didn't really see a big significant difference in size between the queens that were reared in the miticide-free or the miticide, do you say Latin? Laden? L-A-D-E-N. Laden. Wax. Or cups. So, no significant difference in weight. Given from the statistics that I said, if the magic number is 0.05, so if it's higher than 0.05, there's no significant difference. Thoracic width, the queens reared in miticide-free wax were larger as far as the thorax width. But, interestingly, the ones that were reared in miticides had a slightly bigger head. But again, I told you that these morphometrics don't really seem to equate to the other reproductive quality measures. But it's still something good to measure because it's easy. You grab them. Okay, as far as, these are the cool results, very interesting results. As far as queen reproductive health, this is the total number or the average number of alive sperm in a standard volume of 500 microliters. So you can extrapolate that to the total size of the spermatheca, but we didn't do this because it's really difficult to give. Spermatheca is not exactly a sphere. It's a little bit kind of pushed on the sides. So it's hard to say with certainty how many sperm cells there are in the spermatheca. There's only kind of estimates. But this 500 microliter is more of a given amount. So it's better to do it this way. Anyway, as you can see, very interestingly, queens that were reared in the miticide-free wax contain a very, very highly significant, statistically significantly higher amount of sperm, alive sperm, compared to the queens that were reared in miticide-laden environment. And this is the total number of alive cells, sperm cells. This is the same is for the total number generally, both alive and dead. So the total number of sperm cells is significantly higher for queens that were reared in the miticide-free environment, which, using these two pieces of information, you can get sperm viability. And so queens, that's the most important one here. Queens that were reared in the miticide-free environment have higher sperm viability by about 20%. So they have 20% more viable sperm than queens reared in the miticide-laden environment. 80% sperm viability means that 20% of the sperm are dead, basically. Or here, 60% sperm viability means that the other 40% are dead. So there's still not 100% sperm viability, but it's significantly higher than the queens that were reared in the miticide-laden environment. This is very interesting, and we don't really know yet what to make of it. We also expected that perhaps queens that mated with that queens that were reared in the miticide-laden environment would show lower mating frequency, that they would mate for fewer drones. In fact, it was the opposite. So there were queens that were reared in the miticide-free environment mated with fewer drones compared to the ones reared in the miticide containing wax. We do two measurements of mating frequency in biological terms. We do observed mating frequency and effective paternity frequency. They're slightly different, but for us, for our purposes, basically, it's about 24. So queens that were reared in miticide-laden environment had an average of about 24 matings, which is actually high compared to the average. Whereas the ones reared in the miticide-free environment had more like normal mating, like 18 or so. So what to make of that? It can be perhaps that that the ones raised in this miticide-laden environment are needing to make more because a lot of that sperm that they're getting somehow is getting dead inside their spermatheca. Because I showed you that they have a lower sperm viability. So that's the idea that we have right now, kind of overcompensating for the fact that a lot of that sperm that they're getting is dead, so they need to mate with more. But that's just hand waving because we still haven't looked at these physiological questions yet. But it's very interesting. So there are detrimental effects of contamination of wax with the commonly used miticides, fluvalinate, and coumaphos on the viability and reproductive health of queens. Compared to those raised in miticide-free wax, queens that were raised in wax contaminated with fluvalinate and coumaphos had lower total number of sperm cells, lower average number of alive sperm cells, and thus lower sperm viability. And then they also had higher observed and effective paternity frequency, which is curious, although we did not have noticeable differences in body size. So that's something really cool that we're following up on when we start our experiments this spring. Overall, queen reproductive quality influences colony growth. A honeybee queen's phenotype affects other aspects of colony-wide phenotype because we saw that higher quality queens have more productive colonies as far as brood production, comb production, food storage, and population size. In apiculture, beekeepers should always select the youngest possible worker larvae during queen rearing because this shows that it gives you a big kind of advantage if you're using the youngest possible one. There are potentially detrimental synergisms occurring between otherwise therapeutic dosages of miticides inside the colonies, not only in the worker population, but also in the mating health of queens and potentially drones. But overall, we need more studies that look at the effects of queen reproductive quality on procedure, in colony longevity, and so forth. So, to finish off, I want to talk about our research program at Texas A&M and what my research interests are. I am in charge of conducting research 65% of my time. Well, this is just the official assignment, but you learn really quickly that it's 100% of my time doing research, 100% of the time teaching, and 100% of the time serving my department and my community. So these are some of the questions that I am interested in. The reproductive biology of bees in general, the behavioral ecology of swarming. This is a good place, being here in Texas, to entertain questions on the reproductive, the swarming biology of Africanized honeybees, because it is probably different than the things that we've seen with the European honeybee. And we know that Africanized honeybees swarm more often, and they have formed smaller swarms, so this will be really interesting to do similar studies from my PhD dissertation on swarming, but with Africanized honeybees. And we're working on those toxicology and pathology studies with honeybees. We want to help or work in collaboration with other queen breeders in Texas, and at least, or at the beginning, near College Station, to create a better locally adapted queen. I should say that in North Carolina, I was the coordinator of the Born and Bred program for queen rearing and bee breeding. And we trained over 3,000 beekeepers in North Carolina and surrounding states on a very easy way of rearing locally adapted queens using mainly the grafting method, but using other things. And so we have these workshops that if we have someone, we don't have an extension component to our program yet, but eventually in the future, if we get the funding, I would like to create some sort of extension appointment to a person that will be in charge of queen rearing workshops and bee schools and all of these things that I won't be able to do very often because unfortunately I won't be evaluated on my extension endeavors. As much as I love extension, because I've been doing extension for 4 years now, and I really enjoyed it. And pollination of biology of honeybees. Texas is a good place to do studies with cotton, for example. We know that honeybees are not really very involved in pollination of cotton, but few pilot studies from Texas A&M have shown that honeybees do visit cotton, and so that's one of the big pollination ecology projects that we're probably going to get a student working on is cotton. And that's, I'm also going to be teaching, by the way, honeybee biology. Right now it's offered as an online course for undergraduates, but I will be doing an in-person class starting in the spring. But eventually I would love to also have a kind of an extension version of that honeybee biology class for other people other than students to take it in College Station. And I really hope that I will have time to, it won't be very often, but I have some time to come and visit with you and see what your priorities and needs are. And just know that there is now a honeybee biology program at Texas A&M. So with that, I thank you for your time and your
Other questions for Dr. Rangel? We're glad to have you as part of the family.
thank you. I'm really happy to be here.
You're contemplating these studies with biocide contamination for the wax on drums.